Okay, well, welcome everyone to another sterile care webinar. Very happy to have Professor Lori Burrows with us today. For today, I'd like to just start with some opening, obviously welcome remark to everyone on the call. Thank you so much for registering. We are looking forward to a great presentation by Dr. Burrows. Just a couple of housekeeping while we wait for the start. If you could make sure that you are on mute, that would be great. We are recording the video, so this will be sent out to the people who have registered at a very near future. And also, we will be having a time at the end for a Q&A session. So if you could hold your questions for now, Karen and I will be monitoring the chat box as well if you feel more comfortable typing a question in there. Also, to let you know, we are very active, Sterile Care is very active on social media. So if you follow Twitter, LinkedIn, Facebook, Instagram, etc., please join us. We have a lot of updates, a lot of podcasts coming, and a lot of these webinar notifications that come out. So please follow us on our social media. Karen, I'm going to hand it over to you. Karen is going to start with a brief introduction with the company and brief discussion about KiteLock, and then we will move quickly into Professor Burrows' discussion on biofilm. So Karen, having said that, I will pass it over to you.
Thanks, John. Welcome everybody to our biofilm webinar today. It's exceptional to be able to present this the second time due to popular demand. So thanks to everybody for joining. I am going to turn my video off just to make sure that we have good communication. As an infection prevention company with a non-antibiotic antimicrobial, we see great value in educating about this very important topic that is gaining much needed attention in our healthcare practices. But before we hear from Lori, I want to set the stage as to why this talk is so important in the vascular access world. As we know, almost 90% of patients that enter a hospital will require access to their vascular system. Patients with long-term needs, such as chemotherapy, dialysis, and parenteral nutrition, require central venous access devices or venous catheters. So it is our job to make sure these CVADs are free from complications so they receive their life-saving treatment and focus on their already complicated disease. Having said that, the current complication rates in Canada are about 137 infections and 34 deaths per day. Up to one out of 4 patients will die from that infection, not from their disease. And according to the CDC, because we don't collect these numbers precisely in Canada, 1 out of 6 infections are related to antibiotic-resistant bacteria or superbugs. And we know about 22 to 40% of these CVADs have patency issues, which are related to clots as well as biofilm. And unfortunately, we know that cost does lead care. So the amount of money we're spending per year is over around $2 billion a year trying to treat these infections. We don't know exactly what it costs to treat the occlusions. So about 20 years ago, a microbiologist called Dr. Peter Kite out of Leeds, England, took it upon himself to understand what was actually happening inside these CVADs. He used what we call a FAS endoluminal brush that is a wire that has like a Christmas tree at the end, and put it into the CVAD to the tip of the catheter and removed it. When he removed it, he found the tip covered with a slime-like substance. When he put it under an electron microscope, he saw the vast bacterial community. So this is when he validated the presence of biofilm on the inside of a CVAD. We have since learned that biofilm is a source of 80% of CVAD infections. What we also learned is that there's a link between infections and occlusions caused by what we call the triple threat. It is 3 interrelated processes caused by clots, bacteria, and biofilm. Biofilm being the forgotten or misunderstood piece, which is why we have Lori speaking on it today. As you can visually see, the buildup of biofilm and clots puts a CVAD at risk for complications of occlusions and infections. Dr. Kite's work led him to this non-antibiotic antimicrobial tetrasodium EDTA 4%. That's why we called it KiteLock. It is not only able to kill all relevant bacteria and yeast strains found in CVADs, but can eradicate their complex biofilms in under 24 hours of exposure, including the resistant strains such as MRSA and BRE. It is also a trusted anticoagulant, therefore is capable of effectively knocking out this triple threat, resulting in significant improved clinical outcomes. And due to its mechanism of action, it does not contribute to antibiotic resistance. So the last thing I want to speak to is the battle against microorganisms moving forward. When Fleming invented penicillin, he indicated that as soon as we start building antibiotics, the microorganisms will be calculating on how to outsmart them. And as we muddle our way through COVID, it is important that we do not lose sight of the slow-moving pandemic of resistant organisms. What are we doing well now, and what do we need to be doing better? So to understand the severity of this slow pandemic, here's a situation that is on our back door. In India, where you've been able to buy antibiotics over the shelf, people diagnosed with cancer are faced with a decision of receiving chemotherapy and dying of an antibiotic-resistant organism infection versus dying of cancer itself. We no longer can afford to stand back and watch. If we want to be able to provide life-saving treatments, we have to start changing practice now. According to the Canadian AMR report, in 2018, 1 AMR infection was reported for every 180 people admitted to the hospital. For some of these infections, 1 in 5 patients died. Antimicrobially resistant organism infections contributed to about 14,000 Canadian deaths in 2018. The fatal rate of bloodstream infections caused by antibiotic-resistant organisms for VRE more than doubled, for community-associated MRSA-BICs increased by 140%, and 20% of those diagnosed patients died within 30 days of diagnosis. So that gives you some foundation on how this applies to vascular access. Now I'm going to hand it over to Dr. Lori Burrows. She's a professor and a microbiologist, a fellow of American Academy of Microbiology. She's an international expert on the structure, function, and regulation of type 4 pili. I'll get her to say that properly when she speaks. She has published over 120 peer-reviewed papers, reviews, and book chapters. She is currently the Associate Director of McMaster University's Michael de Groot Institute for Infectious Disease Research, and she serves on the editorial boards of the Journal of Bacteriology, the Journal of Biochemistry, and ACS Infectious Disease. So thank you for joining us, Lori. Look forward to your presentation. I will stop sharing so we can hand it over to you.
Thank you, Karen. Thanks for the introduction. I'm just going to share my screen. All good?
Yeah.
Can you see my mouse?
Good for me,
Yes.
Okay,
Yeah.
perfect. All right. So thanks again, Karen. That was a fantastic introduction on the problems of biofilms and antibiotic resistance. So I'm here today to tell you more about what biofilms are and how we deal with them scientifically. So I just want to start with a disclosure statement. So I'm just going to mention some of the research that's going on in my lab in this presentation. And there is a provisional patent on some of the combinations, antibiotic combinations that we're working on. But I don't have a financial interest in any of the other material that's being presented, and I don't receive any compensation from Sterile Care. So today's learning objectives are to help you understand what is a biofilm and how they form, to understand how they tolerate antimicrobial treatment and they enable the development of resistance and the formation of superbugs, as Karen mentioned in her introduction, and to understand physical, chemical, and biological approaches to prevent and treat biofilms. So let's just start off with what exactly is a biofilm? So a biofilm is a community of bacteria growing on a surface. That's the basic definition. And you can find them pretty much anywhere you look in the world. So this beautiful picture of a stream that I have here, if you look at the scum that's growing at the water line along the rocks, that is a biofilm. So it starts off as microorganisms, bacteria and fungi, and then bigger organisms latch onto those, and it grows into this big kind of complicated community. If you looked in the water itself, the density of microorganisms is a lot fewer than what you see attached to surfaces. This is a picture of a not very clean hot tub. And again, right at the water line, you see these communities growing. That's a biofilm. And that's one of the reasons why we put chemicals like chlorine or bromine in hot tubs and pools. And anyone who has a pet will be familiar with the fact that you can feel the slime layer on the inside of their water bowl and food bowl. That is also a biofilm. So it's residue left over from the food or water, and also from bacteria that come from your pet's mouth. And so that's why you have to keep cleaning these things, because they get contaminated with biofilms. And you'll know that biofilms are hard to get rid of. You have to scrub them. They don't just disappear chemically. So Karen mentioned in her introduction that about 80% of infections are related to biofilms. And you can imagine this is particularly a problem with medical devices. So this is a picture from an old review, but I love the way they mapped out all of the different biofilm related infections anywhere in your body. So, starting with sinusitis in the top corner, right? So biofilms growing in the sinus cavities of your head, which, as you know, are very difficult to get rid of. All kinds of medical devices, so stents, artificial joints, peritoneal dialysis catheters, and so forth. Anywhere you can think of, you will find bacteria growing on these things. And although our body is good at keeping bacteria at bay, so your skin surfaces and your mucosal surfaces are good at being antimicrobial, once you break those surfaces with some kind of a line, you allow for bacteria to enter the body. Okay, there we go. So one of the interesting things about biofilms is that they produce what we call a matrix. So they produce this themselves. It's a combination of polysaccharides, so sugars, proteins, lipids, and DNA. And they basically form this matrix around themselves, and it acts like a force field to protect the bacteria. So I have a couple of images here. On the left is a scanning electron micrograph of Listeria, which is a foodborne pathogen, on a stainless steel surface. And when you do scanning electron microscopy, you have to dehydrate your sample, so you can't actually see the matrix very well, but it's this kind of gunky stuff that is gluing the cells together. And if you look at this, a similar biofilm, but under a hydrated condition, so where the water isn't removed from it first, you can see these bright fluorescent bacteria embedded in this blue goo, right? So just like the Matrix, the movie, it's an alternate world that the bacteria build for themselves, right? So they produce this material and it protects them from threats. So let's just look quickly at a typical biofilm life cycle. So bacteria that form biofilms are usually, they start off as sort of free swimming bacteria, and then they eventually come into contact with the surface. And once they get to the surface, they will become permanently attached to it and start to divide and form what we call microcolonies. And it's at this point that they start to produce this matrix material. So that's what I've shown here, is this kind of gray line around them. They will continue to divide and form larger and larger macrocolonies. And I'm showing here just a single bacterium, but this can be really a complicated community of many, many bacteria, many species of bacteria. Eventually, this will get so big that it will either run out of food and oxygen, and the bacteria will release themselves by making enzymes that degrade this matrix, or it'll get so physically big that it'll just break off. I think somebody has their microphone unmuted, so if you can check, make sure you're muted, that would be great.
Yeah. You can help us get an IV in bed 8. Sure.
Connie,
do you guys need it right now?
Earth to Connie, you are not muted.
Oh, okay, that's okay. I'm just in a meeting, but
Let's continue. So why do bacteria and fungi make biofilms in the first place? So I showed you that they, bacteria and fungi like to form biofilms on surfaces, and that's because nutrients concentrate on surfaces. Being in a community like this, where there's multiple species together, promotes communication between the cells. So you can imagine if cells are free floating in a liquid, they communicate chemically, and it's very difficult for them to do that if they're not close together, because chemicals will diffuse, right? When they're in close proximity, that promotes chemical communication between the cells. And those chemical communications can actually activate group-specific behaviors. Bacteria can also transfer DNA easily between themselves, and that can include resistance genes, and they can even transfer cell contents between cells. In a biofilm, bacteria can share what we call public goods. So these are things that will benefit the same species. So you can imagine if they are growing on some kind of surface that they have to break down to eat, if they secrete enzymes, that's a public good, because adjacent cells that are not secreting that enzyme can also benefit when it breaks down the surface. So this actually leads to cheating, because you can have species that don't do anything to help get the food, but they can take advantage of the food. So there's a whole subfield of biofilm biology that is called sociomicrobiology, where people study how bacteria prevent cheaters from taking over the population. And finally, one of the major reasons that they make biofilms is because it will protect them from bacteriophages, which are small viruses that specifically kill bacteria, larger predators, so that can be things like amoeba that eat smaller organisms in your body. That would be white blood cells, for example, immune cells. The biofilm protects bacteria from desiccation. So I showed you that that matrix is normally very hydrated, so that allows the bacteria to retain water in their environment, even when the environment dries out. And then lastly, the matrix will protect the bacteria from antibiotics and disinfectants, because it will help prevent the penetration of those compounds into the biofilm. So before I go on, I just want to mention a few definitions, because I'm going to talk a little bit about this later on. So when we talk about how an antibiotic kills, we talk about things like the minimal inhibitory concentration. So that's the amount of antibiotic that will stop the bacteria from growing. It doesn't necessarily kill them, because depending on the antibiotic, if you take the antibiotic away after you stop growth, they can regrow again. But if you use what we call the minimum bactericidal concentration, that is usually a slightly higher concentration that will actually kill the bacteria, and they will not regrow when you remove the antibiotic. And then lastly, we have something called the MBAC, which is the minimal biofilm eradication concentration. And that is usually even higher than the MBC. So I mentioned that biofilms provide a protection against antibiotics and disinfectants. So if we look back at our biofilm lifestyle map here, the planktonic bacteria, or the free-swimming bacteria, are what we consider antibiotic susceptible. And this is how we actually test bacteria in the lab, ironically, right? We grow them in liquid and we add antibiotics. So the MICs that we're calculating in the lab are usually aimed at killing this type of bacteria. But I already showed you that this type of bacteria in a biofilm is the one that's most often found in the body. So that's a bit of a problem that we need to overcome when we define how we're going to kill a biofilm in a person. So why are biofilms antibiotic tolerant? So I mentioned that there is limited penetrance of antibiotics from the outside into the biofilm to get to the cells, right? So the matrix itself can bind the antibiotic. So a lot of antibiotics have a charge on them, and often the matrix has the opposite charge. So you get charge interactions, and it acts like a filter and just stops the antibiotics from getting into where the bacteria are. Inside this biofilm, this mature biofilm, you have what we call physiological heterogeneity. And that's just a term that means that some cells are growing faster than others, right? So you can imagine the cells at the edge of the biofilm have access to more oxygen and more food, because the food is whatever's in the environment and diffusing into the biofilm, than the guys in the middle of the biofilm. So the ones in the middle, which have less oxygen and food, will grow more slowly. And because most of our antibiotics target things like DNA replication, cell wall replication, transcribing and translating proteins, if you're not doing those things actively because you're not growing very fast, you're less susceptible to antibiotic killing. Being in a biofilm induces stress responses, and we're still trying to understand what these are, but we know that they contribute to the ability of the bacteria to withstand antibiotic treatment. And then lastly, in the biofilm, we have a subset of cells that form, which we call persisters. So if you treat a biofilm with more and more and more antibiotics, you will eventually kill most of the cells, but there is always a subpopulation, a very small number, that cannot be killed no matter how much antibiotic we add. These are almost like spores for the biofilm. And what will happen is after the antibiotic is removed, these persister cells will grow back and the biofilm will return. So the biofilms provide an environment that promotes the development of antimicrobial resistance. So the steps that I just told you about that the biofilm can take allows the bacteria to survive until the antibiotics go away. But because of the environment that the bacteria find themselves in, it actually encourages the development of permanent resistance, not just temporary resistance. So because they're reduced to a lower dose of drugs, the bacteria in the biofilm can acquire mutations that will eventually lead them to be permanently resistant. You get induction of error-prone damage repair pathways, because when you're treating with an antibiotic and you're damaging the cell will frantically try to fix the problem. Sometimes they don't fix those problems very accurately, and that leads to mutations. And some of those mutations are going to end up helping the bacteria survive antibiotics. And then, as I mentioned, because the bacteria are growing in close proximity to each other, they're able to exchange antibiotic resistance genes between cells. So you can imagine if you have a mixture of susceptible cells and resistant cells, and that resistance element is on a transferable piece of DNA, they can swap them like hockey cards in between. And everybody will end up with the same resistance. So I'm just going to mention briefly some of the research that we're doing in my lab, because what we found is that treating biofilms with a sub-inhibitory concentration of antibiotic or disinfectant can actually stimulate biofilm formation, which was pretty terrifying. So we came across this accidentally because we were looking for small molecule inhibitors of biofilm formation that could be combined with antibiotics to improve killing of biofilms. So in other words, if biofilms are tolerant of antibiotics, the idea was, could you find a second molecule that you could combine with an antibiotic that would stop the bacteria from making a biofilm while the antibiotic killed them? And so I had a graduate student working on this project several years ago, and to her horror, instead of finding biofilm inhibitors, she found biofilm stimulators. And it turns out that those were actually antibiotics, but at levels below what can actually kill the bacteria. So I'm just going to briefly show you the type of experimental system that we use. This is called a 96 well peg lid, and it fits into a 96 well rectangular plate. And we really like this system because we can test, for example, 96 different kinds of bacteria, or 96 different kinds of drugs or inhibitors. And what we do is we put the bacteria in the plate, and we put this lid in, and these pegs dip into the bacterial culture, and biofilms will form on these pegs. And then what we do after 24 to 48 hours is we remove these peg lids and we stain them with this purple dye. So the more purple dye you see, the more biofilm you have. And you can see if you look carefully that just like the natural biofilms that I showed you, you tend to get a slightly more biofilm right at the water line, right? Because the bacteria have access there to more oxygen than the guys that are lower down in the plate. So using this kind of system, we can screen lots and lots of different molecules to see what they do to biofilms. And we can either add the compounds and the bacteria at the same time and then try to grow the biofilms. So see if we can stop that from happening. Or we can grow the biofilms first, and then we can plunk this lid, or plunk the lid into the drugs and see if the drugs can disperse the biofilms. So what do I mean by antibiotics stimulate biofilm at sub-inhibitory concentrations? So we have a high throughput drug screening facility here at McMaster, which has thousands and thousands and thousands of compounds, and we can screen them in high throughput fashion. But because we have thousands and thousands and thousands of compounds, we can only screen them at one concentration, because otherwise, if we screen thousands of compounds at multiple concentrations, we would be broke within a month. So we arbitrarily screen typically at 10 micromolar, because that is a dose that is low enough to be potent, but high enough to do something. So if you screen biofilms against a drug like ampicillin, so this is a really common antibiotic that you might get for an infection if you go to the doctor, that kind of antibiotic kills very well. So this organism that I'm showing you in these pictures is listeria, and these experiments were done with listeria. The graph shows you the growth in the plate, because we want to check to make sure if we don't have a biofilm, it's not because we don't have any growth. And in this case, that's true. And the purple is the amount of biofilm that we see on the pegs. So for this particular drug, we saw that at 10, everything was dead. There was no biofilm, but that was also because there was no cells, right? So with no drug, they grow fine, but this is a very potent antibiotic. So because this was a very high concentration for this particular antibiotic, we decided to look at, well, what happens down here? Because we could see that there was a little bit of something happening here at a very low concentration. So we expanded this concentration range over here. And what we saw was really startling to us, that as you go below the minimal inhibitory concentration for killing the bacteria, which is about 0.31 here, now the bacteria can grow again. But as you go even lower, you get an increase in biofilm formation. So we took the PEG at 0.04 and broke it off and looked at it with the scanning electron micrograph. So this is the control PEG, and you can see the plastic is the dark gray in the background. And there is listeria making nice little biofilms all over this surface, but you have gaps between. What we saw in the biofilm that was treated with sub-inhibitory antibiotics, so remember, these are antibiotics below the level that will actually kill them, that stimulated them to make more biofilm. And we just saw an absolute wild mass of cells. And so we couldn't even see the surface. So basically, we're looking at much more biofilm when we treat with antibiotics below the concentration that will kill the bacteria. So it's almost as if these sub-inhibitory antibiotics trigger an anti-antibiotic biofilm response. So we think of this as the bacteria responding, trying to protect themselves because the concentration of the antibiotic is not high enough to kill them. And so one of the things they can do to protect themselves is to make more biofilm to try to keep the antibiotic away from the bacteria. So we're not the only people that notice this. This is another figure from a paper from another group showing the same effect for MRSA. So staph or methicillin-resistant Staph aureus, which is something that is a big problem on medical devices. 2 different MRSA strains at no methicillin, you see no biofilm. But as you increase the amount of methicillin, you start to see huge increases in the amount of biofilm. And so they've done their assays in a slightly different way, but obviously the darker, the more biofilm. And then as you reach the minimal inhibitory concentration for this strain and you kill the cells, then you don't see any more biofilm. So the same idea, though, you see this no biofilm with no drug or very low biofilm. And then as you add sub-inhibitory concentrations of antibiotics, you see spikes in biofilm formation. So it's not just beta-lactam antibiotics, which are the classes that I just showed you. It's multiple classes of antibiotics with all different targets that do this, right? So this is not just a specific cell wall phenotype or an anti-DNA phenotype. So these are examples of other antibiotics. So fluoroquinolones inhibit DNA replication, and we can see, again, at no antibiotic, you have a certain amount of growth and a certain amount of biofilm. As you increase the concentration of antibiotic, the growth does not decrease, but the biofilm starts to go up. And then the growth starts to decrease, but the biofilm goes up even more. So at this low concentration, the bacteria can still grow, but they are provoked into forming more biofilm. We see this with cephalosporins, and we see it with macrolides. So we call this phenotype the, oh my God, I'm gonna die. And then we get high enough concentration and they die. And we see this same phenotype over and over again. We even see this with soap. So it doesn't even have to be an antibiotic per se. It can just be a disinfectant or a soap. So this is a mild soap that's used as a surgical scrub. And Pseudomonas is notorious for being able to grow in dilute soap and disinfectant. And that's exactly what we see here. So again, at 0% of soap, we see a very faint amount of biofilm. And at 100% soap, we've killed the bacteria, so there's no biofilm. But somewhere in between, where we've diluted the soap down below the concentration that will actually kill the bacteria, we cause them to make tons of biofilm. So you can imagine this is happening in, for example, the sinks in a hospital, right? Because people are washing their hands and you have dilute soap going down into the drain. So this organism, again, is notorious for growing in the plumbing in ICUs, for example. So when we do these anti-biofilm screens, we actually found biofilm stimulators that don't even have known bacterial targets. So we screened a collection of drugs that are FDA approved, but off patent now. And we were surprised to find that we saw this biofilm stimulatory effect with lots of different drugs. So again, these don't have an antibiotic effect in that if you look at the yellow bars, they're not really killing the bacteria. The bacteria are still growing, but we see this characteristic increase in biofilm as we increase the concentration of these drugs. So this first drug I'm showing you here, loperamide, this is Imodium. So something that you would take for traveler's diarrhea. Clearly, Pseudomonas does not like Imodium because it's provoking this biofilm response. This is an antipsychotic drug. Again, doesn't kill the bacteria, but we see these spikes in biofilm. So looking at the classes of drugs that do this, it was really interesting to us to see that things that were never intended to do anything to bacteria clearly have an effect on the bacteria. And this was not really well appreciated at the time that they were tested and approved by the FDA, because nobody thought to look at this. So we are not also the only ones to see this. This paper was published a few years ago, and they took the same collection of drugs that we screened and showed that they have a lot of impacts on the physiology of human gut bacteria. So I guess the take-home message here is that when we develop drugs for human uses, we should be aware that they actually can have effects on our bacteria. So this got us really thinking about what was going on here. So the question that we had was, how is this happening? So how is it that sub-inhibitory antibiotics of all different classes can result in stimulation of biofilm formation? So we decided to consider a few different hypotheses, and these are not mutually exclusive. There may be more than one thing going on. So the first one we thought of was, it's possible that sub-MIC antibiotics cause bacterial damage that triggers the induction of prophages, right? So many bacteria have these little bacterial viruses that live in their chromosomes, dormant and quiet, until the bacteria become damaged, because it's kind of like the guy leaving the sinking ship, right? So these prophages don't want to remain in a host that is damaged, because that will be the end of the prophage as well. So when you damage a cell, a bacterium, sometimes that causes the prophages to come out of the chromosome and kill the cell before the cell can die, so they can be passed on to a new cell. So that's one possibility. I mentioned to you that biofilm populations are physiologically heterogeneous. So it's possible that when we treat with sub-MIC antibiotics, some cells are actually more susceptible than others. And so we're killing the more susceptible cells and not the less susceptible cells. And then the debris, the guts of the dead cells, are seeding more biofilm. So they're feeding the rest of the bacteria. And third, we considered that sub-MIC antibiotics might trigger stress pathways. So we know that biofilms are tied in with stress pathways in ways that we don't really understand. But it's possible that when you treat the bacteria with a sub-MIC antibiotic, you are causing damage that triggers these stress pathways. And those stress pathways are tied to production of more matrix and larger biofilm, and that will help protect the bacteria from further damage. So we are continuing to study these three ideas to figure out what's going on. But in the meantime, we thought, I wonder if we can actually exploit this idea, right? So the idea is that if you want to detect the effect of an antibiotic at a concentration that's too low to kill the bacteria, how would you do that, right? So when we're doing, as I mentioned, high throughput screening, and we're screening only at one concentration, that concentration might actually be too low to kill the bacteria. So if we're only asking, can we kill the bacteria, we might miss a few things. However, if we also monitor biofilm formation and we see this increase in biofilm formation, that is the canary in the coal mine that says, hey, this molecule might be an antibiotic if you just dialed up the concentration a little bit. And that will allow us to look for new antibiotics and miss fewer hits. So that's what we decided to do. So the idea is that we're going to screen at 10, right? But we're going to monitor biofilm formation. So I'm giving you the example here of erythromycin. At 10, if we looked at the growth in yellow, we would not call this an antibiotic. Now, we know this is an antibiotic because if you crank up the concentration a little bit more, it's definitely killing. However, if we monitor biofilm formation, you can see that we can already see an increase in biofilm formation well below the concentration that will actually kill the bacteria. So this is our sweet spot for saying, hey, this thing might have some antimicrobial activity. So we did this. We used the same library that I mentioned earlier of off-patent FDA-approved drugs. And I'm just showing you one replicate from some of our screening data here. So these are just two different replicates. And so if they were perfectly correlated, they would form a 45-degree line. And we're looking at what effects these drugs have on biofilm formation. So most drugs have no effect on biofilm formation, which is fine. And then you can see up here, we're getting more and more stimulation of biofilm formation. And a lot of these are antibiotics because, of course, this FDA-approved drug library has antibiotics in it. So we looked at many of these molecules, and one of them really stood out to me, and that's this compound thiostrepton. It's a known antibiotic, but the organism that we screened with, Pseudomonas, is a gram-negative bacterium, and thiostrepton has a reputation for having absolutely no effect whatsoever on gram-negative bacteria. So that was intriguing to me. So why would Pseudomonas object to an antibiotic that doesn't have any effect on gram-negative bacteria? So this is the data that we saw when we pulled thiostrepton out and did our biofilm assay. And you can clearly see that it's stimulating biofilm formation, which is what we saw in our original screen. But this was really a head scratcher to me because this is thiostrepton. It is a huge molecule, so really huge. And we could not understand how this really huge molecule could get into the bacteria because its target is the ribosome, which is in the cytoplasm of the bacteria. And Pseudomonas has a reputation for blocking large molecules from getting into the cell. So I don't have time to tell you the whole story of how we eventually came across how this drug is getting into the cell, but we eventually hit on the finding that this drug was exploiting the bacteria's need to get iron. So Pseudomonas makes this beautiful green pigment, which is this compound pyoveridin, and it makes this pyoveridin molecule to get iron. So it sends it out into the growth medium, and the molecule binds to iron, and then the bacteria bring it back inside the cell, take the iron, and then send the pyoveridin back out again. So this great big antibiotic has somehow figured out how to exploit the pathway used by the bacteria to get pyoveridin into the cell, and it sneaks in the pyoveridin receptors. So this is just some genetic data that shows that the only way you get resistance to thiostrepton is if you delete both of the pyoveridin receptors that this organism makes, then they can grow again. So that was a pretty cool finding, and it validated our idea that you could use this method essentially to find new antibiotics. So we decided to do a little more investigation of thiostrepton as an antibiotic because Pseudomonas is really difficult to kill. And if you could find new antibiotics for this organism, it would be very useful. So because of this link with iron acquisition, we decided to try combining thiostrepton with an iron chelator. And the rationale for this is that the more we starve Pseudomonas for iron, the more receptors for pyoverdin the bacteria make, and the more drug they pull into the cell, right? Because the bacteria think that thiostrepton is a siderophore, is a pyoverdin molecule. And so they basically grab it as fast as they can, especially when there's low iron, because they're trying to get iron. And essentially what they're doing is giving themselves food poisoning. So if you combine thiostrepton, and these are called checkerboard assays with thiostrepton on one axis and an iron chelator on the second axis, you can see that you can synergize, you can improve the killing by the antibiotic when you remove the iron. So this is particularly evident for Diphtheria cerox, right? So the vertical axis shows that on its own, thiostrepton is not that great of an antibiotic, and the chelator on its own is not that great, but when you combine them, you basically wipe out the bacteria. We also tried just putting thiostrepton in the presence of serum, right? So human serum or mouse serum, which is naturally iron limited, and it works great. Even you don't even actually need the chelator, because in this case, as long as you have serum there, you will kill the bacteria. So that was pretty cool. So in the last little bit, I just want to go over some other methods of how we can get rid of biofilms, right? So Karen mentioned at the beginning that kite lock is a chemical method for getting rid of biofilms, but there are also physical and biological methods that I just want to mention. So all of these methods work to some degree, but at the end of the day, it is very difficult to get rid of an existing biofilm. So my favorite example of this is the one on your teeth, right? So we all just had lunch recently, if we're on the East Coast. That stuff on your front teeth, your biofilm, is growing happily because you just fed it. And it's very difficult to get rid of that biofilm. You literally have to scrub it off a couple times a day with your biofilm removal device. Getting rid of that chemically is next to impossible, right? So even when you rinse your mouth with things like mouthwash, you're only killing a small amount of the bacteria and you're leaving most of the other ones behind. So you just have to keep in mind that prevention is better than treatment, because once a biofilm forms, it's really hard to get rid of. So there are a few physical methods that have been attempted to get rid of biofilms over the years. Putting bacteria in an electrical field can actually reduce their ability to attach to a surface. And that's because bacteria are really, really tiny. And so electrostatic repulsion from a surface is a big problem for them, right? So you can actually make that problem bigger for them by charging the surface or the area that they're in. It's not that practical, obviously, for treating people, right? Because it's hard to put a person in an electrical field without causing other issues. Ultrasound is a really time-honored way of getting rid of biofilms. So this is the type of thing that they'll use on, when you take your jewelry into a jeweler and you want them to clean your rings or your earrings, right, to remove the gunk on it, they'll put it in an ultrasonic bath to get rid of things that way. So that's useful for sort of more industrial scale biofilms, but again, a little challenging to do in humans. The last physical method that people have tried is topology. So changing the actual shape or properties of the surface that the bacteria is going to attach to. So I want to give you a cool little example of this. So a colleague of mine, Cynthia Whitchurch, came up with this genius idea for basically tricking bacteria into not migrating along a catheter. So this is all one catheter surface here, but on one side, they have introduced grooves into the surface because bacteria will follow grooves in a surface just the way a person will. Like if you go for a walk in the woods, you don't just randomly walk through the woods, right? You follow a path that someone else has made, and bacteria will do exactly the same thing. If you just give them a smooth surface, they'll go all over the place. But if you give them a groove surface, they will follow it. So I just want to show you this little video because I think it's really cool. So you watch how far the bacteria on the right are able to migrate compared to the bacteria on the left. So this is our favorite organism, Pseudomonas, the one that I was just talking about. And it's really good at crawling across the surface. But you can see if you give them these grooves, they get confused and they start to follow the grooves on the surface instead of just migrating along it. So you can imagine if you put these grooves sort of at right angles on a catheter, that you can have the bacteria just chasing their tails around and around and around the catheter instead of migrating along it and further into the body. So I thought that was a really cool example of using topology to trick the bacteria into not going where you don't want them to go. Okay, so let's talk about this. That was our physical methods. Let's talk about chemical methods briefly. So coatings on catheters has been tried by many people, many kinds of coatings. There hasn't really been a coating, to my knowledge, that is completely bacteria proof. So what happens is if you put a coated surface into the body, molecules from your body will stick to that coating, and then the bacteria will stick to those molecules, right? So fibronectin, for example, other proteins in the blood, once those proteins get deposited on the surface, that's what the bacteria will be binding to. So it's very difficult to make a surface coating that completely resists bacteria. Some surface coatings elute disinfectants or antibiotics, and that works okay for a while, but it just contributes to the problem of antibiotic resistance, right? So unnecessary exposure to disinfectants and antibiotics just makes the problem worse. Lock solutions like Kite Lock are useful because they're not antibiotics, but they are inhibitory to bacterial growth. So they will stop the biofilm from forming in the first place, and they can also release existing biofilms. So this is just a graph from a paper on a Kite Lock study showing in yellow that it can eradicate preformed biofilms after some exposure. So for staph, I talked about MRSA earlier, 6 hours and 24 hours, you can get rid of existing biofilms. And then 2 gram-negative bacteria, Pseudomonas, which is the guy I just showed you crawling along the catheter, and Klebsiella, they're actually more sensitive to this Kite Lock because they have an outer membrane, and the EDTA in Kite Lock will actually strip away the cations in the outer membrane, which is really bad for its stability. Lastly, there are biological methods to manage biofilms, right? So probiotics will establish a community of bacteria on a surface that will basically defend that surface against intruders trying to form on it. Bacteriophages, as I mentioned, are little viruses that are specific for bacteria. They don't do anything to humans. They are actually quite good at penetrating biofilms. A lot of them have developed enzymes on their tails that they use to stick to the bacteria that have the ability to chew through the biofilm matrix, because that's how they crawl through the matrix and attack the bacteria. And lastly, there are enzymatic treatments, right? So the enzymatic treatments that are under development target the matrix themselves. So they don't actually kill the bacteria, but they can dissolve the biofilm. So let me show you an example of that. This is an example from my friend Lynn Howell's lab at U of T. So she studies biofilm degrading matrix enzymes. And they showed that if you add these to an existing biofilm, so this is what's called a pellicle biofilm. So it's just basically a scummy ring that forms at the top of a tube if you just let it sit. This is a biofilm, and if you add these matrix degrading enzymes, you can see that they will chew up this biofilm and the bacteria will just fall out of it. So the biofilm will just fall apart, and this is happening really rapidly. This control enzyme is the same one, but it's been inactivated, so it can't actually chew up the sugars. So it happens really quickly. It's pretty cool. And you can imagine this would be useful for breaking down existing biofilms on surfaces. Okay, so that brings me to the end of my talk. And I just want to leave you with a few take-home messages. So if you can remember, biofilms are the most common mode of bacterial growth, right? And that's because they protect the population from a variety of stresses, right? So anywhere you look in nature, you will find biofilms growing on surfaces. Once they grow, they're very difficult to remove, right? They attach themselves very firmly to a surface and it's hard to get them off, right? So imagine brushing your teeth. That's trying to get biofilm off of the surface. Biofilms can seed disseminated infection. So I showed you when the biofilm gets too big, chunks of it will break off or bacteria will actively leave and go somewhere else. And they also can contribute to the development of antibiotic resistant superbugs because they form a protected niche for the bacteria where antibiotics are less effective. And prevention is key to stopping biofilm formation. So I just want to say thanks to all the talented students in my lab who work on biofilms and our funding sources for money. And then I would be happy to answer any questions.
Thank you, Lori. I'm gonna definitely have to go back through this again to go through it all. You always seem to surprise me with the amount of information you have that is always new. So thank you so much. I'm gonna open the group to conversation. If you'd like to chat on the text, go ahead. If not, please open your microphone and you can ask a question.
All right, I am going to go through the ones that are typed in here. So Dan, do you want to unmute yourself and ask, or do you want me just to read the question?
Hi, yeah, I have a question about the use of heparin as sort of as a lock solution, and heparin is the most biologically negatively charged particle that we're aware of. And could that negative charge somehow have an effect on biofilm if there is an electrical charge to it? Is that working at cross purposes or helping the biofilm?
So it can help a little bit, but the problem is the matrix that the bacteria make is also charged. So you might actually make the problem worse depending on the species of bacterium, right? So the key thing is what exactly is the thing on the catheter that you're trying to treat, right? So the most common catheter pathogens are gram-positives like staph, right? Because that's part of your skin flora. So it's not surprising that if you stick a catheter through your skin, you're going to get skin flora contaminating it. So I guess my short answer to your question would be it depends on the organism. Okay, next question is Deirdre. Okay.
No. Yeah, I
hi.
had one. Hi there. I just had one quick question. Would you recommend antibiotics before we put the catheter in to reduce the biofilm, or antibiotics after?
Definitely not before. So that, we try really hard not to use prophylactic antibiotics unless there's an obvious need for it. And that's because it depends on the length of catheter, how long will the catheter be indwelling, right? So adding antibiotics has the potential for making biofilm worse, as I showed you, right? So if you're giving antibiotics prophylactically, oral antibiotics or IV antibiotics, what's the penetration of those antibiotics at the site of the catheter insertion, right? It's low compared to the load in the blood, in the serum. So unless you're using local antibiotics, which a lot of people will anyways, I don't think it's a good idea just to be adding antibiotics. And we're trying to steward the antibiotics that we have left because we don't have a lot of new ones coming down the pipe, unfortunately. And so if we lose the existing antibiotics to resistance, we're in big trouble. Okay, next question is from Renata. Okay, I'll just read the question. Is there any practical effect over the catheter lumen if I use the pulsatile flushing technique with saline only? probably going to knock off any really loose biofilm, but it would be the same effect as swishing your mouth with water and expecting that you're going to remove all the bacteria from your teeth, right? So if you swish your mouth with water or saline for that matter, you are going to knock off loosely adherent bacteria, but the rest will adhere very tightly. And there are some species of bacteria that we have found the higher the flow rate you subject them to, the tighter they will stick to a surface, right? They will actually adjust their adherence to be tighter under higher flow rates. And that's really, we see that a lot with urinary tract pathogens, that they are responsive to flow rate and they can stick even more tightly. So short answer is you're not getting stuff off by swishing with anything or flushing with anything. Okay, Kieran. Okay, no Kieran. Hi.
Hi, can you hear me?
Sure.
Thank you. Fantastic talk.
Oh, thank you. You got kind of an echo going on there.
Okay, can you hear me still?
Yeah, I think I'm going to read your question though, because there's a funny echo going on when you unmute yourself. So what evidence is there, considering your research info, that we should replace central venous catheters that have been treated with TPA repeatedly? So that's a hard thing for me to answer because I'm not a clinician. And so I don't look at a TPA replacement. Karen, do you have any insight on that by any chance? Sorry to put you on the spot there, Karen.
Sorry, can you explain more the recurrent infections with biofilms, like even when we remove?
No, sorry, this is the previous one. What evidence is there, considering your research, that we should replace CVCs that have been treated with TPA repeatedly?
So I'm not sure exactly what you're asking there, but what we understand is TPA is a thrombolytic, so it breaks down clots, right?
Yep.
So there's a practice in dialysis where they're using TPA as a lock. And there's one study that shows with TPA and heparin that it reduced infections. So I don't know if that's because you're limiting the nutrients for the biofilm that you might see an impact on the infection, because TPA directly, logically, would not have any impact on reducing clots from forming or the biofilm that's sitting there. It would only take the clots away that could be there.
Absolutely. Thank you.
Does that answer the question?
I hope so. Thank you for jumping in.
Yeah.
Okay, the next question is from Sivaranjani. Do you want to unmute yourself and ask? Okay, I will read the question. It says, can you explain more about recurrent infections with biofilms? Like even when we remove the infected catheter and insert a new one, the infection rate is higher. Great question. So depending on where you're putting this catheter, right? So a urinary catheter that gets infected and then is replaced, if the urine contains bacteria from the original catheter, you're going to expose the new catheter to infection at a higher rate. If you're replacing a catheter at a skin site, same thing, right? Around the site of catheterization, you can have a higher bacterial load because of the previous catheter being there, right? You have to remember that catheters are a happy surface for bacteria, right? Your skin is a hostile surface because it has a lot of antimicrobial mechanisms. As soon as we break that barrier with a plastic surface of any type, or a silicone surface or a polyurethane surface, we are providing a route for the bacteria to attach and move up, just like I showed you in that video. Okay. There's a comment that says it's cool that they stick more with a higher flow rate. Yep. Lots of things about bacteria are incredibly cool. Can I speak to the issue of loperamide promoting biofilm formation? Interesting. So one of the things we have discovered about loperamide is that it messes with the bacteria's ability to make ATP in the cell, right? So the bacteria have a membrane that surrounds them, and they have a charge gradient across that membrane that helps them make ATP. I won't get into the specifics of it, but the bottom line is if you add loperamide, you actually disrupt that gradient of electrical ions across the membrane, and that impairs their ability to create energy. So that's probably why they are reacting to loperamide, and that if we just increased its concentration even further, we might damage them to the point where they stop growing altogether, right? But the important thing is that biofilm stimulation effect that we see alerts us to the fact that this molecule that we're looking at might have antimicrobial activity if we get the concentration up a little higher. So it just helps us miss fewer hits when we're screening. Okay, next question, Hannah. Do you want to unmute yourself? Hi.
Yes, hi. So I know quorum sensing is about how bacteria are able to communicate to each other and they release these quorum sensing molecules. So I was wondering like what the research is in terms of quorum sensing with biofilm formation, because I would assume that it would allow them to get close to each other, and whether there's any research in blocking quorum sensing molecules perhaps in biofilm treating.
Yeah, great question. So yes, so when I talked about bacteria being able to communicate to one another chemically, part of that is quorum sensing. So the term quorum sensing was actually coined by a lawyer brother-in-law of a scientist who was studying, like how do bacteria know how many other bacteria are around? And it's because they all release these small molecules that if there's very few bacteria, the concentration of those molecules is very low. But when there are a lot of bacteria, the concentration of those molecules rises very quickly because there's lots of them making those molecules. And so they use those molecules to detect other bacteria in their environment. So if you block quorum sensing, you don't block the ability to make a biofilm altogether, but you do block the ability of the bacteria to make a biofilm that is structured in the way that I showed you. So, it does make them a little bit deaf and blind to each other. And so they don't structure the biofilm properly and in relation to one another. So there are a lot of efforts going on to try to block quorum sensing. It's a little bit hard to do that because there's a lot of different variations on quorum sensing. So antibiotics work on lots of different bacteria because they target common things that the bacteria do or make. Quorum sensing molecules are all over the map, right? So it is difficult to, unless you pick on a specific type of quorum sensing from a specific type of organism, it's a little bit harder to target that. So it is an effort ongoing, and it does have an effect on biofilm formation. It's probably going to be most useful in the long run to target, to add quorum sensing inhibitors to antibiotics, right? So the same idea as the strategy I was talking about that we were taking, which is to try to block biofilm formation so that the antibiotics will then kill the bacteria that are not able to form a biofilm. Okay, hope that answered your question. Fiona, you are next.
Hi, thank you,
Hi.
Dr. Burrows. I was just actually wondering how this relates to sort of lab testing when they report sensitivities, like from a positive blood culture. So we get a report back that says, they've listed all these, the bug, they've identified the bug as well as all the like maybe 6-7 types of antibacterials that they've tested it with, and some will be sensitive and some will be resistant. So, but then how do you know? I'm not sure on the lab side of things, like how do they test it at a kill level for all of them, and if they die then it's sensitive and
That's a great question, right? So I told you biofilms are more tolerant of antibiotics than planktonic cells, but we test in planktonic culture. So that peg lid system that I showed you that we use was actually developed specifically to answer this question. So what happens if we do susceptibility testing on biofilms rather than on just planktonic bacteria? And it's difficult to do that in a high enough throughput that a clinical lab can get through all the testing that they need to do is the short answer. We know that putting bacteria under nutrient stress will often make them more susceptible to antibiotics, right? So the other thing we do is we test MICs in rich media in the lab, but in a host, in a human, bacteria are always under nutrient stress, right? So I showed you that serum is iron restricted, right? So your body tries very hard to keep iron away from bacteria because it knows that it will restrict their growth, right? So we know that under those circumstances, antibiotics actually work better. So what is probably happening is the MICs that we get in rich liquid media are high enough that when we translate that to a dose in a human where the bacteria under nutrient restriction and also being attacked by other things, right, like including your immune system, that it still works out. It's not ideal, and there's not good correlation between the MIC that we get and whether an antibiotic will actually work in a person, believe it or not, but it works well enough that that's the system that we still use. Okay. Next question is from Clara. She says, how about the CVC coated with silver? Oh, silver. So I mentioned that people have been trying for a very long time to find the Teflon of coatings that will stop bacteria from sticking to a catheter, and they have not been able to find that. So silver is one of the ones that does work reasonably well. Silver kills by basically damaging membranes and molecules. So it's a very non-specific type of killing, and it's actually a very old type of killing. So the Romans used to put silver coins in their milk to keep it fresh because it would kill bacteria. So, we've used silver, we've used copper. So copper pipes in plumbing also kill bacteria for a long, long time. But what we're starting to see is silver resistance, because so many people are using silver for everything now, right? It's in toys, it's in your socks, it's in your underwear, it's in your gym clothes. The more we put these antimicrobials out into the world, the more pressure we put on bacteria to resist them. So short answer is it does work to a degree, but at the cost of encouraging further resistance. So if you need to use an antimicrobial catheter because the patient is in rough shape, then you can do that. But it's always better to use a plain catheter if you can get away with it, because then you don't encourage resistance unnecessarily. Okay. Dan, you have another question. Do you want to read that?
Yeah, thank you. Hi there. Yeah, so a colleague of mine has been horsing around with using alcohol and sodium citrate in various concentrations, and certainly we see 70% being effective. So how does, have you looked at ethanol as something as a biofilm disruptor, or does it? Any comments?
So yes, it will work because it will dissolve membranes, but that's, I always say, the best antimicrobial is bleach, but I wouldn't want to actually have it anywhere near my my catheter. So things like ethanol can kill bacteria in biofilms, but it doesn't disrupt the biofilm, right? So it's not going to solve the problem of occluding the catheter, right? It'll kill the biofilm in place, and then you'll just be left with a big clump of dead bacteria. And then where's that stuff going to go? So you have to think about that. We do think about, what is the cost of disrupting a biofilm, depending on where it is, right? So, my favorite example is on artificial heart valves, right? If you get vegetations or biofilms on an artificial heart valve, and you give the patient something that will basically dissolve that biofilm, you can cause an embolic shower, right? If you disrupt that biofilm too suddenly and chunks break off and end up in small capillaries, you can give that person a stroke. So depending on where the biofilm is, you have to be careful about disrupting existing biofilms as opposed to trying to prevent biofilm formation in the first place. So that's just something to keep in mind. Ruth.
Yes, I just wanted to, if you could comment, hi, on we see a lot of people using antibiotic lock solutions when they have catheters and they don't want to remove it. Just wondered if you could comment on that.
So do you mean, so they've already diagnosed that the catheter is infected with a specific organism and they're trying to just keep it patent?
Yeah, they're trying to save it for maybe someone's had, they need the line for long-term use. They've had multiple catheters and they keep getting infections. So then they try and save this particular one by locking it.
Yeah, so I would say that that's probably, that is a case where you may have to use antibiotics, but it's important to know that the bacteria growing in the biofilm on that catheter are going to be more difficult to kill, even with an antibiotic that has been, tested against the organism for the reasons that we just talked about earlier, right? So when you send that organism for culture, the way they test it is not the way it's actually growing in the catheter. So as a lock, you can add a very high concentration of antibiotics. So you could probably get around that, right? Because you can add an antibiotic at a concentration that you couldn't necessarily give the patient because it might cause side effects. So that would be an example where you might get away with it. Okay. Lots of thank yous. You are welcome. Okay. Viona has another question. Hi.
Thank you. I was just wondering if the thiostrepton is available for clinical use in patients, and would it be indicated to use in combination with an antibiotic for a patient who has this, repeated CLABSIs or a suspected infected clot?
So great question. I wish I could say yes, but the answer is no. So it's currently not approved for use in humans. It is approved for use in veterinary indications. The problem with thiostrepton, even though it's a fantastic antibiotic, is it's not very soluble. So that has limited its commercial development, right? So it's, I think I talked to a couple of people in the antibiotics industry and they said, oh, thiostrepton, that stuff is brick dust. In other words, it's something that they don't like to work with because it's very difficult to manufacture and formulate in a way that you can use it in a human. However, that's why we're quite excited about the finding that we have that if you reduce the concentration of iron in the environment, you actually can use less of the antibiotic, because the more starved the bacteria are for iron, the more they suck up thiostrepton. So you can add less of it, which kind of overcomes the solubility problem. So we are actually in the midst of doing mouse experiments right now to show that if you combine thiostrepton with an iron chelator, which is already FDA approved, that is an effective combination, and the solubility is not a problem. So hopefully in the longer term, we'll be able to use it for things like catheter locks or actual antibiotics. I hope. All right, that's the last question that I see. I'm happy to stay on if anybody else wants to ask any more questions.
Lori, I'm curious, when we talk about antibiotic locks, so many times we hear not to use them because they will promote resistance. So is that a fear that you would still have? What would be your choice, to not use them unless you absolutely have to? Or
so yeah, I would not use it for an initial catheterization, right? You don't need to, really. It should be reserved for the cases, and I'm saying this as a non-clinician, right? This is just my experience in the biofilm field. If you can get away with not using an antibiotic, that's the route that you should take. If you're stuck with a difficult patient that's had multiple issues with their line and you really need to save it, then that's a case where I would say it's justified, right?
Right.
we always, I just want people to think really hard before they they grab the antibiotic as their first choice, right? Because as I said, we are running out of useful antibiotics, right? We're starting to see organisms now that are resistant to every single antibiotic that we have, right? And that is terrifying, right? I mean, COVID showed us what happens when you get a pathogen for which you have no drugs, right? You have no defense against these things except for public health measures, right? So we don't want to get there with bacteria.
So
prevention, yeah.
Yeah, it would appear, Lori, that your best line of defense would be to prevent from the start. Is that correct?
Yes, exactly.
Well, you're always a wealth of information. I learn something every time I listen to you speak. So if there's no other questions, I cannot thank you enough for taking this time and helping educate on such an important subject.
No problem. I could talk about biofilms all day long.
I could listen all day long.
Yeah. Thank you, everybody, for joining us.
Yes. Thank you so much.
Thank you.
Any additional
Have a great
questions,
day.
I would be happy to field those as well.
All right. Awesome.
Thanks so much, Lori.
Great. Have a good day.
You
Thank
too.
you.
Thanks, everyone.
Sean, you'll be getting a copy